Welcome to the comprehensive guide on Bacillus species. In this video, we will explore the characteristic and pathogenic potential of these fascinating bacteria. Before we dive into diagnosis, it's important to understand the basics of Bacillus genus. A key feature that makes them unique is their ability to form endospores, allowing them to survive extreme conditions. While many species are harmless, some like Bacillus anthracis are highly pathogenic and of major veterinary importance. So let's begin with the overview of this important bacterial genus. Whether you are a veterinary professional, microbiologist, or a student, this video provides a complete overview of the Bacillus species. General features of Bacillus. Bacillus species are large, gram-positive, endospore-forming rods. Although older culture may appear gram-variable or even gram-negative due to spore formation, usually Bacillus are gram-positive. Most Bacillus species are aerobic or facultative anaerobe, with the exception of Bacillus infernus, which does not form spores in oxygen. They are classified into several groups like Bacillus subtilis group, cereus group, Bacillus circulans groups. In that, Bacillus subtilis group include Bacillus subtilis, Bacillus licheniformis, and the second, the Bacillus cereus groups, which include Bacillus anthracis, the causative agent of anthrax, as well as Bacillus cereus and Bacillus mycoides. And the last group, the Bacillus circulans groups. Bacillus species are catalase positive, generally grow well on nutrient agar but not on Macongia agar, and are mostly motile except for the Bacillus anthracis and Bacillus mycoides. Most are mesophilic. Notably, Bacillus anthracis is a significant pathogen of herbivores and humans and is recognized as a potential biological weapon. So, the general feature of Bacillus that we should remember is it is a gram-positive endospore forming rods, which is aerobic usually, and catalase positive, and most are motile except Anthracis and mycoides. Natural habitat. To understand how Bacillus species impact human and animal health, we first look at where they live. Generally, Bacillus species are saprophytes, meaning they thrive in the environment, especially in the air, soil, and water. Their secret to survival lies in the spores, which are incredibly hardy. These spores can resist heat, radiation, and even harsh disinfectants, allowing them to remain viable in the soil for decades. While Bacillus anthracis spores are geographically ubiquitous in soils worldwide, often infecting grazing herbivores, there is a key distinction in their life cycle. The vegetative form of Bacillus anthracis is only found within the host. It does not typically exist in the environment. This highlights its nature as an obligate pathogen in the case of Bacillus anthracis, whereas species like Bacillus cereus and Bacillus licheniform are often just opportunistic pathogens. Pathogenesis and pathogenicity. Bacillus anthracis is a major pathogen responsible for anthrax, a highly fatal disease affecting herbivores, mammals, and humans. In humans, anthrax occurs in three major forms: cutaneous, gastrointestinal, and respiratory anthrax, depending on the route of exposure. In animals, the disease is typically peracute to acute, characterized by massive septicemia. The bacteria multiplies rapidly in the bloodstream, producing potent exotoxins that lead to severe hypotension, shock, and sudden death. Death occurs primarily due to the action of lethal toxins, which disrupt cellular signaling pathway and cause widespread tissue damage. Because of the peracute nature of the disease, animals often die suddenly, leaving very little time for clinical diagnosis or treatment. In systemic infection, the mortality rate is animals approaches 100%. Species susceptibilities varies. Cattle, sheep and goat are highly susceptible. Horse and humans are intermediate susceptible species, and these pigs, birds and carnivores are considered resistant even though they can be infected when exposed to high doses of the organisms. Anthrax has also been reported in wildlife including bison, white-tailed deer, ostriches, mink and moose. Rats and some strains of mice show strong resistance to infection. This variation in susceptibility plays a key role in the epidemiology and control of anthrax outbreak. Diseases in animals. In animals, anthrax shows marked variation in severity depending on the species affected. In high susceptible species like cattle, sheep and goat usually develop the peracute form of the disease. This form is characterized by septicemia, high fever and sudden death, often occurring within 1 to 2 days of infection. Postmortem examination reveals characteristic findings such as tarry blood from orifices like mouth, nose and anus. A hallmark feature is the failure of blood to clot along with the incomplete rigor mortis. And the spleen is markedly enlarged, soft and dark, commonly described as blackberry jam spleen. In less susceptible species, the disease presents differently. In horses, the anthrax commonly cause subcutaneous edema of face, throat and neck and may be associated with colic also. In carnivores, the infection usually results in gastroenteritis following ingestion of the contaminated meat. In pigs, anthrax occurs sporadically and is characterized by throat swelling with or without digestive signs and is typically associated with a low mortality. In this slide, we will learn about the different species of Bacillus. First one is Bacillus anthracis. Its primarily animal host is cattle, sheep, horse, pig and carnivores. In ruminants, that is the highly susceptible species, it will cause peracute or acute systemic infection that will result in sudden death in those affected animals. In equines, it will cause colic, enteritis or localized swellings, and in swine, it will cause pharyngeal swelling or intestinal distress. And about the impact on humans, this Bacillus anthracis is a zoonotic infection. It can cause 3 forms of infections in humans. First one is cutaneous, gastrointestinal or respiratory or inhalational anthrax. The second species is Bacillus cereus. It will affect cattle, dog and cats. In cattle, it will cause occasional mastitis, and in the pets like dogs and cat, it causes rare instances of food-related toxicity. And humans, it is a common cause of foodborne diarrheal illness. Next species is Bacillus licheniformis. Its primarily animal host is cattle and sheep. It will result in reproductive issues like abortions and mastitis in ruminants. And in humans, the same like food-related gastrointestinal distress. The last one is Bacillus subtilis. It is also affecting ruminants like cattle and sheep. And it is associated with again abortion and bovine mastitis. And in the case of humans, it is considered low risk and rare opportunistic issues. Next is its transmission. Transmission occurs mainly through ingestion when grazing animal consumes soil or feed that is contaminated with the anthrax spores. The organism can also enter through skin abrasion leading to cutaneous anthrax. Inhalation of spores is less common in animals but remain an important route, especially under certain environmental conditions. During outbreaks, biting flies may act as mechanical vectors contributing to the spread of infection among animals. Once inside the host, the endospores are taken up by the macrophages where they germinate into vegetative bacilli. These bacilli are then released into the bloodstream where they multiply rapidly, leading to septicemia and toxemia. The virulence of the Bacillus anthracis depends on 2 main plasmids. First one is plasmid PX01 that encode a tripartite anthrax toxins, while plasmid PX02 encode poly D glutamic acid capsule, which is strongly anti-phagocytic. The anthrax toxin consisting of three components usually, that is protective antigen, edema factor and the lethal factor. When protective antigen combines with the edema factor, it forms the edema toxin causing tissue swellings and fluid accumulation. When the protective antigen combines with the lethal factor, it forms the lethal toxins which disrupt cellular signaling, lead to cell death. Both capsule production and toxin expressions are tightly regulated by the host signals, such as increased carbon dioxide levels, ensuring maximum virulence inside the host. Together, these mechanisms explain the rapid progression and high fatality rate seen in anthrax infections. Molecular virulence factors of Bacillus anthracis. The pathogenicity of Bacillus anthracis is primarily driven by the two large virulence plasmid that we discussed in the previous slide, that is PX01 and PX02. Together, these plasmid enable the organism to evade the host defense and cause severe systemic disease. First, we will learn about the PX02 plasmid that play a critical role in immune evasion. This plasmid encodes the synthesis of poly-D-glutamic acid capsule, which forms a protective outer layer around the vegetative bacterial cells. Unlike the polysaccharide capsule, the capsule is poorly immunogenic and effectively prevents phagocytosis by host immune cells, particularly macrophage and neutrophils. As a result, the Bacillus anthracis can survive in the host bloodstream for a long period of time, and it can multiply, contribute to the persistent bacteremia and septicemia seen in Bacillus infection. The presence of PX02 plasmid is therefore essential for its full virulence, allowing the organism to resist immune clearance due to its low immunogenicity and help to establish infection. The capsular virulence of Bacillus anthracis is controlled by a cluster of genes that is encoded by the PX02 plasmid. The first set is cap A, B and C. It codes for the poly-D-glutamic acid capsules, which act as a shield that prevent macrophage from engulfing or phagocytosing the bacteria. The next is the DEB gene. It encodes the capsule degradation factor, which play a role in controlling depolymerization of capsular material. This process is important for the capsule remodeling and regulation during infection. Finally, the ACP8 gene functions as a key regulatory element, controlling both capsule expression and capsule assembly in response to host environmental signals. Together, these genes ensure efficient capsule production, regulation and maintenance, making the capsule a major determinant of immune evasion and virulence in Bacillus anthracis. So, PX02 plasmid mainly involved in capsule formation, and it helps in immune evasion by preventing phagocytosis. Next, we move to the plasmid PX01, which encode the potent toxin that cause the clinical symptoms of anthrax. These are divided into two main complexes. The first one is the lethal toxin. It composed of lethal factor and the protective antigen, and the lethal factor is encoded by the LEF-A gene and the protective antigen encoded by PAG-A. The lethal factor is a zinc metalloproteases that inactivate MAPKK, leading to the cell death and the release of inflammatory cytokine and triggering oxidative burst in macrophages. The second complex is the edema toxin or EDTX. This consisting of edema factor and again the protective antigen, and the edema factor is encoded by SIA and the protective antigen is PAG. Edema factor acts as a calmodulin dependent adenylate cyclase, which increases cellular cyclic AMP levels and disrupt water homeostasis, leading to the characteristic swelling that we seen in this disease. Note that in both cases the protective antigen acts as a binding modality, essentially serving as a key that facilitates the entry of the lethal and edema factor into the host cell cytoplasm. Next, the regulatory genes. Finally, the Plasmid PXO1 containing several regulatory genes, most notably ATXA, serves as a master activator of transcription for the toxin gene, along with the PAG-R and TOP-A that ensure the bacterial virulence factors are expressed at the right time during infection. Together, the capsule from PXO2 and the toxin from PXO1 make the Bacillus anthracis a highly effective pathogen. Next is the sporulation process. The sporulation occurs in carcasses under aerobic nutrient-deprived condition, allowing bacteria to survive harsh environment. The process begins with asymmetric septation, where a vegetative bacillus or mother cells divide unevenly to form a smaller phosphor compartment. Next, the phosphor matures, developing specialized protective layers that make it highly resistant. Then the mother cell dies, lysing to release the mature spores into the environment. Finally, the endospore production is complete, with each mother cell yielding one resilient dormant endospore capable of surviving extreme conditions. Structure of Bacillus anthracis endospore. It's a highly resistant dormant survival form of the bacterium. As its cause, it containing DNA, essential enzyme, calcium and dipicolinic acid, which help stabilize proteins and DNA. Surrounding the core is the cortex. It is a thick layer of peptidoglycan that provides structural support. Next is the spore core, composed of multiple protein layers, giving the spore remarkable resilience to chemicals and heat. The exosporium is the outermost layer containing BCLA. It's a Bacillus collagen-like protein of the anthrax, which play a role in host interactions. Chemically, the endospore composed of protein, DNA, calcium, dipicolinic acid, paracrystalline lipid layer, and cross-linked peptidoglycan. It containing very little or no water and has no ATP, carries out no metabolisms, making it an inert but highly durable form capable of surviving extreme conditions. Germination of the Bacillus endospore begins when nutrient molecules such as germinants bind to the specific receptors on the spore surface. This triggers the release of calcium dipicolinic acid from the spore core, followed by an influx of water which hydrate the core. Next, the cortex lytic enzymes are activated, finally breaking down the cortex layer. Finally, the spores transition into a vegetative cells ready to multiply inside the host and resume normal bacterial growth. Besides Bacillus anthracis, several other Bacillus species are important in veterinary and public health. The first one is Bacillus cereus, can cause food poisoning in humans with diarrheal or emetic type, and occasionally affects dogs and cats. Bacillus licheniformis is associated with bovine abortion and mastitis. Bacillus subtilis can cause food poisoning, mastitis and ovine abortion. Bacillus coagulans cause bovine abortion. Bacillus pumilus is associated with bovine mastitis. Bacillus thuringiensis is an insect pathogen, is widely used as a biopesticide due to its crystal toxin genes. Other related pathogens include Penibacillus larvae, the cause of American foul brood in honeybees, and Clostridium pyliform, formerly known as Bacillus piliformis, is responsible for the Tysos disease in mice and foals.